Hello students, welcome to Vedantu NEET English. I am Ms. Gopika, your own biology master teacher. I hope I am audible, visible and the presentation right behind me is also visible. Please let me know in the chats if we are live yet. Quickly let me know if we are live yet and can we start the class. I am also checking. Yes, we are live. Very nice. Yes, we are live, right? Hello. Sorry for making all of you wait for some time, okay? Yes, can we start? Today what are we doing? We are doing the important concepts of biotechnology principles and your processes, okay? So biotechnology, we can actually complete the whole chapter, but the thing is, see, when I'm trying to explain you, when I'm trying to make you understand every small bit of it, it is going to take some time, okay? Hello, good evening to all of you. I hope all of you had a great, day yesterday. Yes, now I can see your chats. Okay, all of you quickly like the video and share the video with your friends and ask them to join as well. So shall we start? Biotechnology principles and your entire biotechnology unit is a very important unit. Most of the time from both the chapters you will get around 8 questions. Okay, so around 8 questions you can expect, and this also has a good weightage, okay, 36 to 40 marks you can expect from your biotechnology. Every year here and there small variations will happen, but still it's a good high weightage chapter, okay, good high weightage chapter, okay. So shall we start? What are we going to understand? So students, what is this biotechnology? When we talk about biotechnology or when we talk about this whole thing, we have all learnt about biotechnology. Traditional methods like making curd, okay, it is one of the biotechnology method, but it is a traditional biotechnology method. Now in this chapter you're going to learn about what is modern biotechnology method. Like I tell you, if I want, let's say, give me a name. All of you give me one name. Okay, I think I have to inform on Telegram, students, because I changed the time. So just give me 1 second, okay? And let me ask them to join on Telegram as well. My kids are so lazy, they won't open and see. I have to tell them. Okay, I have to tell them, please join, kids. Your exam it is. You're going to be doctor. Yes, done. Okay. Okay, done. Ready, all of you? Yes. So students, as I told you, we have already learned the traditional biotechnology methods, be it making curd, be it making your toddy, many techniques we have been using, but we did not know that we can do much more than this, or our ancestors did not know that we can do much more than this. And right now we are in a situation where we can do a lot with biotechnology. And let me tell you, if you like this chapter, you can also go into this field, okay? You can go into this field, okay? Yes. Very nice, very nice. Okay, so look here, biotechnology principles, if I talk about, there are mainly 2 principles mentioned in your NCERT. So here I am not going to talk about EFB's, definition, what is traditional, modern biotechnology, not at all. I am directly going to talk about the principles. So 2 principles are given in your textbook. Can all of you tell me, do not oh, today also board is dancing, students. Last time when I took biotechnology also board was dancing. Okay. Yes. So, principles of your biotechnology. What are the principles of biotechnology? See, I want to make a modern biotechnology. I want to make improvements in biotechnology. Now, let's take an example. Let's say there is a hen, okay? Hen, and the name of hen is Sam, okay? Hen's name is Sam, okay? And let's say this hen is very hefty, okay, very hefty, and but even though it is very hefty, it does not lay enough eggs, okay, it lays less eggs, okay, it lays less eggs. And let's say there is another hen, okay, another hen, let's call the hen as Poma, okay, Poma. So these two hens are there, Sam and Poma. Now Poma, even though the Poma is not a very hefty hen, it is a very, we looking at the hen, we feel like, oh, this hen is going to die now, like very tired hen, okay. So this is not a very hefty hen, but it lays more eggs. It lays more eggs. Understood? It's not hefty, okay. Now let's say with the biotechnology, I want to make a product. Okay, I want to make a product which has, okay, the hen I want to make. Okay, the hen I want to make is Zen. Zen is the hen's name. This Zen should be hefty so that I can, sell it for KFC and other places, hefty. And at the same time, it should be laying more eggs. Now, ma'am, you have too many wishes, ma'am. Like my students have wishes, sleeping that I should get 720, I should get 680 without attending class. Like that only when I want to make the Zen a more, a very like perfect hen. How? Ma'am, you have one hen which is hefty but lays less eggs, and then you have another hen who is thin and not very strong, not very, looks very tired, but it can lay more eggs. Now, what do? With the help of genetic engineering, with the help of biotechnology, I can take the desired gene from Sam and Poma and make my Zen out of it. Can you imagine? Okay, Zen out of it. Clear to all of you? Yes, is this clear? Is this clear? This is a very important concept. That's why I am asking you over and over again. Okay, this is the whole chapter itself. It's the introduction for your chapter. So to make Zen, you can take the desired gene of Sam, which makes the hen hefty, and you can take the desired gene of Poma, which allows more eggs production. So this way, I will get an egg which is hefty and also produces more egg. This is awesome, ma'am. Right? You're not at all selfish. You made something out of it. Now, how does this principle work on? How does this whole process happen? So the principles of biotechnology are two things mentioned in your NCERT. First word used is genetic engineering. What is it called, students? Genetic engineering. Okay, and the second thing that they have mentioned is bioprocessing. Mentioned is bioprocessing. Yes? These are the two principles, yes or no? Look at your NCERT and tell me. This is the thing, right? Okay, I'll just reduce the brightness, okay? I think the brightness is too much. Better now? Okay. Yes, it's a little more better, right? Yes. So, students, this is the two words used. Now, ma'am, what is this two words meaning? Right, what is genetic engineering? This is what is genetic engineering, where I am able to make a recombinant DNA, where I am able to take a desired gene, insert it somewhere and do something out of it. So here, being genetic engineering is where I am able to form a recombinant. Okay, recombinant. I am able to use a recombinant technology or a recombinant DNA. Okay, clear? And then what is bioprocessing? I have never heard about it. Bioprocessing means, let's say I took a desired gene from Sam, I took a desired gene, and I took desired gene from Poma, I inserted into the host and I told you please make this. You please make this, I want Zen. Now, okay fine, the gene is like, okay, I will take up the responsibility, but you need to make sure that I am in a sterile condition. The environment around me is so sterile that I will be the only one that is growing and I will not be damaged. No other source should damage me, because as I told you, in a lab when you are making many things, okay, let's say you are growing or you have kept your culture plate, but there is another person who has brought fungus into the lab. No problem in growing that, but the problem is if the spores scatter, then all the culture plate will get contaminated. So we have to make sure that whenever we enter the lab, we are in a sterile condition, including we sanitize our hands, the lab coat, hair, everything should be tied up. Clear to all of you? Okay. Okay, now look here, students. So genetic engineering and now bioprocessing, as I told you, it is where we should be able to maintain a sterile condition. Okay, sterile condition for our desired gene to grow, and this sterile condition should also enable, okay, it should also enable the growth of whom? Enable the growth of only desired gene. Is the principle clear to all of you? Yes or no? Yes, sterile condition has to be maintained. Very good. Okay, all of you are understanding? Perfect, perfect. Okay. Now, students, when we talk about genetic engineering or when we talk about the whole concept in this chapter, we many times use the word gene. But do you know what's a gene? This is a DNA, right? This is a DNA strand. A small segment on a DNA is called a gene. Yes, ma'am, I knew this. Okay, some of you might know this, some of you might not know this. A small segment on the DNA is called a gene. And this will be able to express, or this will be able to code. That is why I am calling it a gene. All of you remember structural gene we studied in molecular basis of inheritance. That is the gene that is has the capability to form the mRNA. Like this gene is a small segment of a DNA. Okay, remember these words. It's a small segment of DNA, which can also code. Okay, which can also code. Clear? Yes, Sangeeta Sagar is telling something. Now, let's say I have taken the desired gene. Okay, I have taken the desired gene. Now, my task is I have to make sure that the desired gene enters into the host. So, let's say students, this is the gene that I have taken, a desired gene I have taken. Okay, from the DNA. Okay, one small portion of the gene I have taken. That gene has to be, it has to enter into my host, right, so that I get what I want. But if I want to allow this gene to enter into my host, there should be a carrier or there should be a vehicle, right? Who is the vehicle in this? Who is going to carry the gene for me till the host? Okay, who is going to do that job for me? All of you tell me in the chat box. Who is going to allow me to put my desired gene with, you know, to allow the entry of my desired gene into the host? All of you in the chats, quickly tell me. Yes, all of you are capable of telling because we have studied this. Okay, we have studied this. Yes, quickly. Yes, Vishnupriya, very, very, very correct. What is it? Ma'am, you need a carrier, or in other words, you need a vector. A vector means what? Now in malaria, when I taught you, I told you that mosquito is the vector. Now how did I tell mosquito is the vector? I told mosquito is the vector because it is carrying the disease causing, disease causing plasmodium. So I told it is a vector, and sometimes there are cases where vector itself has one life cycle. Now as we saw in mosquito, right? Now same way here also, to transfer this desired gene into the host, I will need a vector. I will need whom, students? A vector. Clear to all of you? This is easy. So this is your introduction to biotechnology. What is biotechnology? What are you going to learn? All that you are going to learn is how to construct a recombinant DNA technology. What are the tools required? What is gene cloning? What is gene vector? All this is what you are going to learn. Okay? Very good for those students who answered, but I am not at all, happy about the other students who are not attending class regularly. Okay, I'm very disappointed in them. Yes, so shall we continue? Now, the main steps that involve in your recombinant DNA technology. So today I'm going to take all the tools of the recombinant DNA technology. I'll give you an overview, and after that, if required, we will do one more part of this. Otherwise, this is good. Okay. Students, so far any doubts, all of you tell me in the chat box. Perfectly okay. Only I can see Madhumita being okay about it. Rest of you? Okay, very good. Okay. Now when I have to do this gene transfer, okay, when I have to do this genetic engineering, there are few things that I need to know. Okay, genetic engineering, when I need to do, I have few steps that I have to remember. The first thing and the foremost thing is I should know how to construct, okay, how to do construction. Not building a home or a villa for someone and giving, okay, construction of your recombinant DNA technology, or sorry, recombinant DNA. Construction of recombinant DNA is my first task to understand. Second thing is how I can do gene cloning and how I can do gene transfer. So these are the main three things that you need to know when we talk about genetic engineering. But don't you think the terms are very easy for you to understand? Yes or no, the terminologies are very easy. Look at it. Construction of recombinant DNA, gene cloning, gene transfer. Doesn't the terminology sound a little more easier for you? Yes ma'am, I can understand that okay, I need to form the recombinant DNA technology. I have to clone this gene inside. I have to transfer this gene for something to happen. This is the whole process, right? Now students, when we talk about this, don't we have to know who was the first people to ever discover recombinant DNA technology? So the first people to discover recombinant DNA technology was Stanley Cohen and all of you in the chats. Now they'll tell ma'am we don't know. I'll give you a clue. H, okay? Not working. B, okay? HB. HB. Try to tell HB. Some boy or something like that. Yes, very good. Herbert Boyer. Okay. Herbert Boyer. These two people in the year 1972 came up with your recombinant DNA technology. Okay. Recombinant DNA technology. When? In 1972. Yes, this is easy to remember. Yes ma'am, it is easy to remember. These were the two people who came up with the recombinant DNA technology. Okay, understood. Now what is this exact recombinant DNA technology? Ma'am, from that time you are telling one thing. Okay, now let's take an example. Okay, let's take an experimental setup. Okay, let's say this is your desired gene. Okay, this is the desired gene that you have. Okay, this could be the desired gene from Sam, or it could be the desired gene from your Poma. Okay, Poma or Sam. This is the desired gene. Okay ma'am, understood. Now this desired gene I am planning to take us like this. This is the entire content. Okay, Sam, this the entire DNA molecule of Sam is there. From that I am going to take a small desired gene out of this. Okay, I am going to take a small desired gene out of this. This is the desired gene that I removed, right? A small segment of my DNA. Now the next part of this is, one second students, board is acting very nice today, special, this is my desired gene. Now you will have a question, no? Ma'am, how did you actually chop this desired gene, or how are you going to put this desired gene into the vector? How are you going to do all of this? So students, it's not that easy like I'm talking about. If I want to remove a desired gene from anywhere, I will need scissors, right? I will need scissors. Can I take a spoon and be like, okay, you take this much of recombinant DNA, this much of desired gene? No, I will need something called as my restriction endonuclease. So restriction endonuclease is going to do the job of chopping this gene and giving it to me. Okay, who is that? Your molecular scissors. Molecular scissors. They are the one who are going to do this job of chopping and giving it to me. Now, okay, chopping done. Now, ma'am, we need a vector, right? We need a vector. Without a vector, will the thing happen? Let's take E. coli. Okay, let's take E. coli. All of you are answering very nice. Let's take E. coli. This is your E. coli. In the E. coli, genetic material, inside that there is an extra small circular extra chromosomal structure which is called the plasmid. What is it called? Plasmid. From this, I am going to remove the plasmid, who is going to act as my vector. So the most commonly used vector is whom, students? Plasmid. What is plasmid? Extra chromosomal circular DNA molecule is my plasmid. Okay ma'am, you removed your plasmid, understood. Now what are you going to do with this plasmid? To this plasmid, I am going to add this desired gene. I am going to tell that plasmid, you are my vector. What is your job? You have to go up to the host and enter into the host cell and give me what I want, right? I want it to be in the progeny. So this desired gene will be added here with the help of someone called as my DNA ligase. What is it going to be, students? DNA ligase. Okay, DNA ligase is going to what? Ligate, glue my desired gene into the plasmid, and finally I will get my plasmid which will have the desired gene. Is this clear to all of you? This is just the basic I am telling, and I am assuming that you have done this chapter once. Okay, once you have done the chapter, so this will be easy to understand. Now students, I am adding the desired gene. Okay, desired, I think I should add it in the other side where I drew the DNA ligase, but this is not correct, so I will draw it this side. Okay, yes, I got my desired gene inside the plasmid. Now what is my task? Now you tell me what is the task. Yes, very good, joining. Now the task is to make sure that the plasmid enter into my host cell. Okay, so we are taking a host cell. This is my host bacterium. I think if I draw with the same color, my students will think that I took it from there and I put it back there only. Okay, yes, this is my bacterium. To this bacterium, I am going to add this plasmid. Okay, I am going to add this plasmid which has my desired gene. Okay, desired gene. Clear? This is what is recombinant DNA technology. Clear to all of you? This is what is recombinant DNA technology in simple words. Okay, now this is my host, right? This is my host. So here what has happened, students? Recombinant has happened, right? This got recombinant. So very simple. We are going to take a desired gene. Okay, now students, let's take an example of your pancreas. Okay, insulin, because insulin is very easy for all of you to understand because you have studied it in human physiology. So here let's take pancreas. Okay, in the pancreas, who is going to make the insulin? Ma'am, islets of a set of cells are there, a group of cells are there which are called islets of Langerhans. Islets of Langerhans. In the islets of Langerhans, we have beta cells which are releasing insulin. Yes or no? This we know. Now I want the bacteria, let's say E. coli, to make maximum insulin for me. For that, what I will do? I will go to the beta cells, nucleus, take the DNA, I will take the gene that is forming the insulin, cut the gene and add it into my host. Simple as that. Then insulin production will happen. So during that process, what are the things that I need? Very good, islets of Langerhans. Okay, so I am going to take the insulin. This insulin forming gene, right, is going to become my desired gene. It's going to become my desired gene. Now this desired gene, I can put it into the plasmid or a vector and insert it into the host. Host is going to do rest of the job. So far good students, this I have done it, this example I gave so that you will understand. It's very relatable for all of you. Clear? All of you? Yes? Okay, assuming that you understood, okay, because your replies will come a little late for me. Yes, now we will continue into the depth. Yes. Very good. Very good, very good. Understood? Yes. Now students, steps of recombinant DNA technology. There are 3 steps given in your NCERT, right? 3 steps given in your NCERT. So we have to learn that step as it is. Okay. First is going to be identification. Okay. First is going to be identification. Identification. Now students, 3 words are given in your textbook. Identification of DNA. Okay, identification of DNA with the desired gene, because imagine you want to make insulin. You don't even know from where you are supposed to get the gene. Let's say I want to make sure that I am getting a hefty hen which lays more eggs. If I do not know which is the hen that is hefty, which is the hen that is going to give more eggs, where am I going to get the genes from? So first job is going to be identification of DNA with the desired gene. Clear to all of you? Easy peasy. With desired gene. Okay, clear? This is the first step. All of you open your NCERT and check. Is it correct or wrong? Okay, obviously it's going to be correct. Second one is introduction. So I understood that I know that I'm going to get this DNA, okay, this gene out of this DNA. It is clear for me. Now I have to make sure introduction of DNA within the host. The last or the most important step is maintenance. Okay, maintenance of the introduced gene into the host, as well as it has to get transformed into the progeny, right? Yes or no? Very good, very good, very good. Sneha, Vishnupriya, Madhumita, Rani, awesome. So maintenance of my transformed, or let's say maintenance of my desired gene and transfer to the progeny. Okay, transfer to the progeny. Yes or no? So this is your 3 steps that are involved in recombinant DNA technology. Okay, yes, very good. Transfer into the progeny. Identification of the DNA with desired gene. I should know that what is the desired character I want out of this and where do I find it. Okay, then next is going to be introduction of the DNA within the host. The desired gene has to be introduced inside the host. Then I have to make sure that I maintain it properly till it gets transferred into my progeny. These are the 3 steps. Clear to all of you? I don't think it is very difficult. There is no rocket science. It's simple. Okay, introduction, first identification, then introduction, then maintenance. Now when we talk about recombinant DNA technology, when we keep telling about this, we have to understand that there are some tools for our entire recombinant DNA technology to happen. But how did this whole thing happen? There is a finding of each of it. Okay, every tool is very, very important. So let's see the tools of recombinant DNA technology. Rules of recombinant DNA technology. Okay, tools which are used. Okay, these are the things that I need to use no matter what, so that I get what I want, right? I want to get my desired gene, for that I have to get. Okay. Okay, now look here, tools of recombinant DNA technology, students. First tool that I am going to use is called as restriction endonuclease, which my students already answered because my students are not studying it for the first time. Okay, restriction endonuclease. My second tool that I am going to use is going to be my DNA ligase, which you have studied in molecular basis of inheritance. What is the third tool that I am going to use, students? It is going to be my vector. Yes or no? Vector should be my third tool. Yes, I can see the answers already. Restriction enzyme, polymerase enzyme, DNA ligase, cloning vector. Very good. Okay, I should also know about DNA polymerase, who is going to make a new template for me. And also I should worry about the host as well, right? I should also worry about the host to whom I have to enter this desired gene. Correct? Yes? Very good. Okay. So, restriction endonuclease, DNA ligase, vector, DNA polymerase and host. In that, even if you skip DNA polymerase, not a problem. But these 4 are very, very important for you, because you should know that what is this restriction endonuclease? What is DNA ligase? How are they formed? How did this whole invention happen? Okay. So, can we go to the invention? Okay. Can we go to the invention? So, students, first we are going to pick up one tool at a time. Okay, one tool at a time. First tool is going to be your restriction endonuclease. Restriction endonuclease. Before I go to restriction endonuclease, my students have to know few more things. Okay, yes, competent host, that we will go into detail later. Okay, students, before, you don't have to tell me that you have a doubt, just tell the doubt. Okay, because I do not have time to see if you have a doubt. No, just tell the doubt, no problem. Now students, listen carefully, that when we talk about your restriction endonuclease, there are few things that you have to remember. That is, Ma'am, how did the discovery happen? Definitely that is a question. But before that, you should know what is nucleases. What is nucleases? So nucleases means, from the terminology itself you understand, something to do with the nucleus. So these are enzymes. These are enzymes that cleave the nucleus. Enzymes that cleave the nucleus. Clear to all of you? Enzymes that cleave the nucleus are your nucleases. Now, two types of nucleases are there. Two types of nucleases are there. Endonuclease and exonuclease. Endo means what? Inside. Exo means what? Outside. Endonuclease means, let's draw one diagram to understand that. Okay, let's draw one diagram to understand that. This is, let's say, one gene here and one gene here. Okay, here also let's draw it with a different color. Here we have one. Okay, here we have one. I'll draw it there later. Okay, 1 second, students. Here also I'll draw. Yes. Okay. Okay. I was drawing some. Okay, look here. Endonuclease and exonuclease, when you are talking about, yes, very good. Exo, your first we will talk about endo. Endo means it is going to cut at the center or within, okay, within the gene. Got it? It can cut within the gene. So this is important for all of you. Endo means within or inside. Okay, inside, within the gene. So here you can see within the gene it has cut and center it has made a mark. Exo means what? It is at the end. Okay, exo means at the end. So at the end. Clear? So here you can see same thing has happened. At the end they have cut. Okay, at the end they have cut. So these are the two ends and these are within. So this is clear. Endo and exo is clear. Endo means inside, exo means outside. Okay, now nucleases is clear. But don't you have a doubt ki, ma'am, how did this whole thing happen, or how did they find about your restriction endonuclease? Anyone wants to know about it? Yes, exo, they cut away from the center. So we can tell like that, or else you can tell it the other way round. Okay, other way round. No doubts, I hope. Perfect, perfect. Okay. Now, students, try to understand that when we talk about the discovery of your restriction enzymes, we have to understand that there was a finding. Okay, finding. In that finding, what happened exactly? That your E. coli. Okay, is everything fine? 1 second. Yes, sound is fine only, no students? Okay, now look here. Let's say when your E. coli, okay, this is your E. coli, okay. E. coli was affected by a bacteriophage, okay. You know, E. coli bacteriophage conversation is very easy for us to remember because we always talk about Hershey and Chase experiment. And what is a bacteriophage? Those who do not know bacteriophage, raise your hands in the chat box. Bacteriophage, okay, bacteriophage, some people tell bacteriophage, you can tell bacteriophage or bacteriophage is a virus that infects bacteria. Virus infects bacteria. So, ma'am, why are you telling about this? So, students, when a bacteriophage, okay, was affecting the E. coli, for E. coli self-defense, it made restriction enzyme. which were able to cut the genetic material, because what do bacteriophages do? They will release the genetic material into the E. coli, then we cannot do anything about it, right? We cannot do anything about it. That's why virus is very dangerous. But what did E. coli do? E. coli used restriction enzymes which can cut, okay, molecular scissors to cut the genes, and with that it also released methyl groups as a self defense. Clear to all of you? This discovery is clear. Any doubts so far in this? No, right? Perfectly clear? Yes, very good. Okay, so as a take of self defense, okay, as a take of self defense, the E. coli formed your restriction enzymes which were capable of cutting your DNA. Okay, cutting the DNA. Why? Because the bacteriophage is going to release the genetic material. Okay, restriction enzyme, and also the part of it, it also released methyl groups. It added methyl groups again as a part of their defense. Done? Perfect? Yes, very good. Okay, as a matter of self-defense, this is how restriction enzymes was found, and this is how your bacteriophage, like E. coli, got protected from your bacteriophage, so that whenever they release their genetic material, they can cut them because of the restriction enzymes. Done? Done, all of you? Clear? Yes, perfect. Okay, this is your self-defense. So this is the finding. Okay, this is the finding. Now, further, when we go and understand how this restriction enzymes cut, okay, cutting of the restriction enzyme or functioning, or how do these people really work, we understood that this enzymes or your restriction enzymes have a technique to cut, okay, action of restriction enzyme. One technique is that neither they cut in the center, okay, they cut in the center and they form the blunt ends, or they cut at the a little away from the center, okay, that forming the sticky ends. Now all of you look at this, this is two strands of it, okay. Let's say this is from 5' to 3' and this is from 3' to 5'. Okay, same thing here, 5' to 3', 3' to 5'. Okay, these are your two strands, clear? Now I am telling you that your restriction enzyme can cut in two ways. Let's say the first way is cutting right at the center. So look here students, T, C, T, G, T, C, T, A, A, G, A, G. So same thing I have done. So here now the font is not the same, that's why, let's say exactly here they make a cut, okay. This becomes a separate part, this becomes a separate part. Okay, same thing goes here, that side also the same thing is going to happen. This will lead to what? This is going to lead to blunt ends. Blunt ends. Now here, okay, here let's say they can also make sticky ends. What can they make, students? They can also make sticky ends, and if they are making the sticky ends, it is not going to cut at the center, okay. It is not going to cut at the center. They are going to make sticky ends in such a way that, let's say here you have, you know, like this, it is given TC. You have to see, if you look at this example, we can take this example only because it will be easier than this. So the example given here is GAATTC, okay, given in your NCERT. Now for that, what do you write? We can write it reverse. Again, palindromic sequence I have to teach you. AATT, okay, C. Okay, this is the sequence. Now, when sticky ends cutting is going to happen, what is going to happen? They are going to cut in a different pattern, and the pattern is as follows. Okay, the pattern is going to go where I cut off the G, okay, G, and this whole thing comes with me to one side. Okay, one side, and this side comes this way. So let's separate it, and if we separate it and write, what are we going to get? We are going to get G, C, T, T, A, and we are going to get this side. So if I have to separate, I have to write it more fast, students. A, T, T, C with G down. Clear? This is the sticky ends that I got. Completely separated. Now you will have a doubt. Ma'am, you told that, okay, restriction endonuclease is there, restriction enzymes that they are going to cut. But do they cut at any random spot? No, they do not cut at any random spot, students. You have to remember that these people are going to cut at a specific recognition site or recognition sequence. That means if the restriction enzyme, okay, restriction enzyme is going to cut, it is always going to cut at what sequence? It is going to cut at a recognition sequence or a recognition site. Recognition sequence or a recognition site. Now, how is this recognition sequence or recognition site identified? Ma'am, how do I identify the sequence? Very simple, students. It's called as palindromic sequence. What is it called? Palindromic sequence. Now, what is palindromic sequence? What is the example given in your NCERT? Palindromic sequence means just like your Malayalam. Okay, Malayalam, that means from whichever way you read it, okay, Malayalam, you read it this way or you read it this way, you're going to get the same spelling. Let's say other word is mom. You read it this way, you write it this way, it is going to be the same. Same thing, let's write wow. Okay, wow. Read it this way, read it this way, the same. Okay, these are called as palindromic sequence. Okay, yes, amma. Okay, amma, madam. All these are palindromic sequence. However you read it, A to A, see AMMA, AMMA, both are going to read at the same way. Okay, same way. Clear to all of you? Yes, what is Tanish telling? Okay, perfect. No doubts, right? All of you are super clear on it? Yes, thumbs up. So this is your palindromic sequence. Now, depending on the enzyme, depending on each of the restriction enzymes, they are going to have a recognition site for themselves, right? So all your restriction enzymes are going to, all restriction enzymes will have a particular recognition sites. Okay, recognition sites. Clear? Clear? Now, depending on the palindromic sequence, you are going to make the cuts. Okay, so you will have two types of cuts, like I mentioned. One cut where you cut exactly through the center, not you, the restriction enzyme cut exactly through the center. That will give us blunt ends. What ends? Blunt ends. Okay. On the other side, it is cut a little away from the center. Okay, away from the center. And this will form your sticky ends or cohesive ends. Okay, cohesive ends. Clear? This is super clear, students? Clear to all of you? Yes. Clear. Fine, fine, fine. Okay. Yes. Perfect, perfect. Okay. Yes. No doubt so far, no? So far, no doubts? Okay, now I'll be able to see the chats. That time I was not able to see the chats from here. Okay, shall we move on? Shall we move? Okay, fine, very good. Now students, we'll try to understand something called as how do you label, what is the nomenclature of labeling your restriction enzymes. Okay, we are going to take a big, we will write it big. Okay, we are going to take HIND 2. Okay, still bigger we should write. HIND 2. Now there's a way to label. You cannot just randomly get up today morning and be like, oh, you are 𝐂𝐴𝐴1, you are 𝐂𝐴𝐴2. It did not come like that. Every letter in that has a meaning and we have to know how the nomenclature has happened. Okay, how the nomenclature has happened. So students, H stands for the genus name, which is here Haemophilus. Okay, which is the genus name. Now here IN stands for influenzae, Haemophilus influenzae, all of you remember from pneumonia. So here IN stands for species and D stands for the strain. Okay, what strain is it? Now in 𝐂𝐴𝐴1 you will study that the strain is RY13. Okay, here what is it going to be? The strain. Okay, RD or anything, strain, depending on which strain it is, we will give that letter for this. Okay, now the 2 recognizes. Okay students, the 2 here stands for when, depending on when the discovery happened or order of discovery. Okay, so in the strain D, the first or the second restriction enzyme which was found was HIND 2. Okay, so depending on the order of discovery. Okay, order of discovery. Clear? This is the nomenclature, how you are going to label your restriction enzymes or how you are going to name them. Okay, name them. Same way in your textbook 𝐂RY is given. So that also all of you can do. Right, 𝐂R1. Now each of the letter, can you tell me the indication one by one, all of you? What is E standing for? Is it a genus or a species name? What is 𝐂 standing for? Okay, what is R standing for? And what is 1 standing for? Okay, like this you have to be in a position to tell. Okay, because 𝐂RY and your HIND 2 is given, so it is easy for you. Yes, go one by one. Tell me what is E standing for? It is the genus name. Very good. It is a genus name. What is the genus here? What is the genus here? Yes, very good. Very good, Madhumita. Very good, Rani. Very good, Roshni. Yes, ma'am. It is genus name which is 𝐂𝐡𝐤. Okay, and 𝐂 stands for your species name, coli, 𝐂𝐡𝐤 coli, and R stands for the strain RY. And in strain RY, the first ever identified one is your order. Okay, yes. So this is going to be okay, this is going to be order. Okay, order of discovery, which is going to be your 1. Okay, in this case it is 1. Done? Is this clear to all of you? Nomenclature is clear, right? All of you, it is very, very clear. So this is how you are going to name your restriction enzyme, and that is what we call as nomenclature. Okay, nomenclature, or how you name the restriction enzyme. Name the restriction enzyme. Okay, clear? Easy? Easy. First genus, then species, then strain, then the number. Okay, order of discovery. Okay, have all of you marked your attendance? Let me check now. Yes, all of you marked? Very good. Okay, all of you marked. like the video if you are enjoying so far and if you are understanding so far. Okay, shall we go further? What is the second tool, students? So we have understood about restriction endonuclease, that is nucleases means something that can cleave the nucleus, right? Two types of nucleases is the endonuclease and exonuclease. Endo means which cuts within, exo means which cuts at the end. Then we understood that how did the finding of restriction enzymes happen. Restriction enzymes, what are they? Molecular scissors, right? So when E. coli was affected with bacteriophage is a virus that infects bacteria. E. coli as a self-defense started producing restriction enzymes which were cutting the genetic material so that the bacteriophage genetic material does not, affect E. coli. Second defense mechanism that they did was to release methyl groups, again as a self-defense technique. Okay, that was the finding of your restriction enzymes. Now, other than that, what did we learn? We learned that if there is a restriction enzyme, it can cut in two ways depending on the sequence that they have. And the sequence or the recognition sequence or the recognition site is the site at which the restriction enzymes will cut. Okay, it will not cut at random spot, it will cut at a palindromic sequence, that is a sequence that is read the same way from front to back and back to front, right? And it is also written the same way. So that is called as palindromic sequence, like example like Malayalam, Amma, Mom, Wow, Madam, etc. Now, depending on how they cut the palindromic sequence, two types of ends we can get. If they are going to cut right at the center, they are called as blunt ends or flush ends, and if they are going to cut somewhere away from the center, they are going to form sticky ends or cohesive ends. So, sticky ends or cohesive ends are like ready to mingle type of people. Okay, now ready to mingle means what? Single, ready to mingle. That means why do people tell it so funny way? Because sticky ends are sticky, they will be like, okay, I need to make sure that I go and attach somewhere. So that sticky ends have high chances to go and attach faster. Okay, so ready to mingle type is your sticky ends. Blunt ends are not like that. They're like, I'm too yard, I don't want. Okay, like that type of people. So blunt ends and sticky ends. Now we understood that if there is a restriction enzymes, there is a way to name the restriction enzyme. That is, the first letter indicates the genus, the second two letters, the next two letters indicate the species, the third letter indicates the strain, and the last is the order of how it was discovered. Okay, so Gopika Ma'am expresses over. So far understood what we did? Yes? So much is clear? I hope so far no doubts. Okay, so we have understood about the first tool. Okay, first tool of your recombinant DNA technology. Okay, recombinant DNA technology. What is the next tool? Ma'am, so simple, DNA ligase. The next tool is going to be DNA ligase. What is DNA ligase, students? Ma'am, I know that they are going to ligate something, right? Ligating, gluing. So these people are your gluing enzymes. Gluing enzymes. So they'll make sure that, let's say, from this gene, okay, from this DNA, you took a desired gene. Okay, let's say you took a desired gene, which is this. Okay, this is your desired gene that you took. Now this desired gene will be taken, okay, will be added into your vector, which is a plasmid, right? A vector which is a plasmid. For gluing of this, we will use it so that the desired gene are glued with your plasmid, right? So here who is going to help us? DNA ligase is going to help us. Okay, DNA ligase is going to help us. So here DNA ligase, here DNA ligase, and this is why we call them as the gluing enzyme. Okay, DNA ligase. Yes, good. Or if you drink some water. Yes, it's like a gum, right? It's like a gum. Okay. Okay. So this is clear. DNA ligase is clear. Now, students, Then next, in DNA ligase only this much is there, okay. Thank God you do not have a lot, okay. Otherwise my students will be like, ma'am. Now students, when I taught you biotechnology, I taught you 3 different types of restriction endonuclease, type 1, type 2, type 3. So if you want a detailed video, definitely check out Biotechnology and Principles by Gopika Ma'am. You will see part 1 and part 2. But here this is an overview, okay. So I'm not going to every single topic. Definitely revision as a whole chapter will be done later. But right now I'm only picking out on some topics, so you will go through it, okay, refresh through it. Now once you understood DNA ligase, you have DNA polymerase. DNA polymerase is going to do what, students? So let's say from DNA polymerase are going to do the job of forming the new template, right? New strands or your new template is going to be formed by your DNA polymerase, correct? So that is your DNA polymerase. Again, I am not going into the depth of DNA polymerase. We will learn it differently, separately, okay. Next thing I want to tell all of you is, I want to know the DNA fragment I have taken, okay. I have made it into fragments. I have to understand the size, okay, of the fragment. I have to see the fragment. All this I want to do. So for that, there is a topic in your NCERT which is called as gel electrophoresis. All of you remember gel electrophoresis. What is gel electrophoresis? Until all of you tell, I will draw the diagram. Tell me, what is gel electrophoresis? I don't want it cross. Yes, this is your agarose gel, right? What is your gel electrophoresis, students? Now look here, this is how gel electrophoresis is going to happen. Okay, here there are going to be wells. What do I call this? Wells, a little deep portion where I can add my DNA samples. Now to this wells I will add my samples. Now on this well, what ma'am, on this entire gel electrophoresis, what is there? Inside this, students, we have agarose gel. What gel? Agarose gel, which is extracted from your seaweed, extracted from seaweed. So on the agarose gel, there are wells which are made at a particular depth, okay. To this wells I am adding my DNA samples, okay. I am adding my DNA samples. Now say I have added my DNA samples, what are they exactly going to do? Remember that your DNA is a negatively charged. DNA is negatively charged. If it is negatively charged, it is going to run towards the positively charged. Yes or no? So here also same technique we are going to use. Let's say positive charge here. The negative charge DNA is going to run towards the positive charge. But how is the running going to happen? Now all of you know, if the fragments are larger, they are going to run slowly, right? If the fragments are thin, smaller, they are going to run faster. Right. So here also same technique is going to happen, students. The fragments which are large will be in the further place. That means they are going to be closer to the wells itself. Okay. Let's say these are the fragments. Okay. They are going to be here. Okay. They are going to be here. But the fragments which are small are going to run faster and they are going to be further away. Clear? Further away. Okay. Clear to all of you? Okay, very nice. Okay, very nice. So here your larger fragments are going to be there. So remember this way, that your larger fragments will run shorter distance, right? Larger fragments are going to be nearer and your smaller fragments are going to run farther. Okay, smaller fragments. Smaller fragments. This is your agarose gel. This is your agarose gel. That is basically the principle behind gel electrophoresis is the size of the fragments. Okay, size of the fragments. So here I am giving you an introduction to your gel electrophoresis. Okay, gel electrophoresis. where you have to understand the principle of gel electrophoresis is the principle that based on size, the separation is going to happen. The separation will happen. That means, what do I mean? I mean that smaller fragments will move further and the larger fragments. Students, all these notes will be available on Vedantu NEET English Telegram, and if you want to join my Telegram, it's Biology by Gopika, okay, as you can see the name on the top, okay. Both on my Instagram and Telegram is the same name, so you can connect with me there. So students, smaller fragment, larger fragment will move nearer. That means it's going to be closer to it. Now this is the principle of it, okay. Now let's say I want to see the fragments on the agarose gel, okay. I want to see the fragments on the agarose gel, right? Yes, undigested, digested will be there. We will not talk about that. I think you would have studied it in Principles of Inheritance a little, okay, where you have the undigested and the digested, okay. Yes. So students, here now I know that my agarose gel, this is my agarose gel, okay. I'm trying to draw 3D and all for all of you. Perfect, okay. This is my agarose gel, which does not look like agarose gel because this end is not there. Yes, this is my agarose gel. These are the wells. These are the wells. Now, after the wells, I have got some bands as well, right. I have got some bands. I have got some smaller bands as well, okay. This is my positive sign, okay. Anode, cathode is there. Now students, I want to see this agarose gel. For that, what am I going to use? I am going to use a dye, okay, which is called your ethidium bromide. So from this gel, I am going to add, okay, ethidium bromide, which is, see, as soon as I add ethidium bromide, it will not be visible for me, okay, but it is a dark orange dye. Now, I have to make sure that I see it under the UV rays, okay. I observe it under the UV rays. I will be able to see bright orange fragments. Okay. So I will use ethidium bromide under UV rays. I will be able to see a bright orange color. Now, if I want to separate the DNA fragments, okay, then I have to do a process which is called elution, okay, not your dilution, okay, elution. That is where I will be separating my DNA fragments. Clear? Separate DNA fragments. Perfect. Is this clear to all of you? Any doubts? Yes. Under the UV light, I will be able to see a bright orange color. Good job. Good job. Okay. Yes. Very good job. Very good job. Okay. All of you are understanding. That is making me so much happier. Okay. That all of you are understanding and answering also very well. That is more important. No, that all of you are answering more. Right. Very good. Okay, elution is clear, separation of your DNA fragments. Okay. Now students, one question I saw, the question was that, is DNA separated? Okay, DNA fragments, are they separated because of free diffusion or is it because of heat or light? So if such a question come in NEET where they ask you that DNA fragments are separated according to which among this, and they give light, heat and free diffusion, please do not answer that. Please understand that with the help of free diffusion, some separation will happen, but will it exactly give me the correct fragment size? It is not going to give me, okay. A small amount of free diffusion is going to happen. Very small amount of free diffusion is going to happen. But it is not going to give me it is not going to give me the actual size of my fragments. Okay, so free diffusion is not your answer. At the same time, is there any light and heat or light that is going to give me the separation? Of course not. No light and heat is going to give me any separation. So these are the 2 points that you have to remember. Please remember this separately. In case question come, that day you shouldn't get confused. Okay, totally the gel electrophoresis or this, the movement is happening because of electric field. Because of electric field. Clear? So your answer should be electric field, not by free diffusion, not by heat and light. Okay? Done. Gel electrophoresis is clear. No doubts about it, no? So here also I have given you a small understanding of agarose gel, larger and smaller fragments. There also I have written on the principle. Okay, I have written the principle. Clear? Yes. Next thing is, students, what is the next most important topic that we are left with? Okay, what is the most important topic? That is your vector. So there should be a characteristic for an ideal vector, right? A characteristic of an ideal vector should be there. Let's say, okay, I'm going to draw something. Yes. Now tell me, all of you, what should be the characteristic of an ideal vector? Ideal vector. First of all, what is vector? Vector is nothing but a carrier or a vehicle. A carrier of vehicle. What are they carrying? What are they transporting? Okay, they are transporting the desired gene for you, right? And the most common vector, okay, the most common vector is your plasmid. Is what? Plasmid. Clear to all of you? Most common vector is plasmid. Now, what is a plasmid? As I already mentioned to you, a small chromosomal circular DNA. A small chromosomal circular DNA. Okay, very good, very good. All of you are answering. I'm so happy that all of you are answering. Now, students, what should a vector have? What are the ideal characters a vector should have? Ma'am, as a vector, what it should have? Maybe it should have something called as a cloning site. Maybe it should have selectable markers, or it should be easily available. It should also be, connecting with the host. That means we have to check that the host that we are using and the vector is in sync with each other. So can you all tell me one each? Okay, one each. Okay, I can see some answers from Chitti. Okay, Chitti was telling about plasmid. Okay, tell me. First of all, they should be able to self-replicate, right? Otherwise, what is the use? So they should have the ability of self-replication. Okay, self-replicate. Ability of to self-replicate, and especially the self-replication, when we talk about, we should talk about our ORI site, that is origin of replication site. That is also very necessary for this people. Okay, yes, must be self-replicating. Very good. Second thing that they should have is, they should have a link with the host. That means they should be at a position where we can introduce them into the host. Otherwise, what is the use? If they are a threat to the host, we are not supposed to introduce them. So it should have a link with the host, or should be suitable for the host. Okay, or instead of link, you can use the word suitable to the host. Clear? Second thing, okay, or the third thing is that they should have cloning site. They should have cloning site. And the last one is they should also have selectable markers. Okay, at the same time they should be easily available, which also comes with this. Yes, they should have selectable markers. Selectable markers. Is this clear to all of you? What is the characteristic of an ideal vector? Okay, vector. Clear? Cloning sites must be present. Very good. Selectable markers should be present or should have selectable markers. Yes, perfect. All of you are able to answer. That means all of you are revised once at least in your life. Okay. Yes, once at least in your life you have visited biotechnology. Okay, vector is clear now, a carrier or a vehicle. Most common vector is a plasmid. What is a plasmid? It's an extra chromosomal circular DNA which is present inside most of the bacteriums like a E. coli. Now, if I draw students a E. coli, see if I draw an E. coli, this is how your plasmid is going to be, okay. This is your E. coli. Okay, give me some time to draw the E. coli. Yes, this is the E. coli, okay. Inside the E. coli, we will have the genetic material. With the genetic material, we are going to have a plasmid which is an extra chromosomal circular DNA. Clear? That is your plasmid, okay. Diagram done? Yes. Selectable markers we will come into, okay. We will come into, I think all of you are answering already. Are you all enjoying the chapter? Is there any doubts so far? Okay. Now, look here students, this is one of the vectors, vectors that you have to study, okay. In this vector, you will see a lot of things. Now, what is each of these things we are going to understand. But before that, do you see here that they have first of all an ORI site? See, according to the characteristics of an ideal vector, first thing that they should have is they should be self-replicating and they should have an ORI site. And in this vector, we have an ORI site. So, first checkbox is done that they can be self-replicating. So, students, if we put a desired gene, okay, near the ORI site, it should also favor replication, right? Because it's closer to the ORI site. ORI site is an origin of replication site. ORI is origin of replication, okay, origin of replication. Origin of replication site should be self-replicating and as well as it should be able, if I put a gene there, it should be available, it should make sure that it replicates. Now, the second terminology that you learned was that cloning site. Now, what is cloning site? Cloning site is a site that suits or it allows cloning to happen. Okay, what should happen students? That facilitates cloning. I will use the word facilitates cloning. Now, identify the cloning sites here students. Can you identify the cloning site? All these are cloning site and we should make sure that the cloning site on the vector, there should be only one cloning site on the vector. Like in the sense, I am trying to say that everything should be different. All the cloning sites have to be different, okay. Repetition shouldn't be there. Now, SAL1 is there, SAL1 cannot be here, okay, on the same vector. So, every vector will have a different cloning site, okay. So, here you have SAL1, BAMH1, okay, HIND3, CLAV1, ECORI1, PVU1, PST1 and let me tell you PBR322 is very important for you. Diagram should be understood. Diagram should be learnt. Okay, diagram should be learnt. Got it? Yes or no? Clear to all of you? Yes. Okay, so you have SAL1, BAMH1, ECORI1, CAL1, ECORI1, PVU1, PST1. Okay, now next thing, okay ma'am, cloning site we understood. Cloning site is something that is going to facilitate the cloning, okay. Now, the next thing I wrote is it should have selectable markers. What is the selectable markers? Students, selectable markers have a job of identifying, transformant and non-transformant or recombinant or non-recombinant type of cells. So it will identify transformant and non-transformant. I am writing NT, and recombinant and non-recombinant. Okay, non-recombinant cells. Okay. So the job of selectable marker is mainly identification. Yes, very good. Select the transformant and non-transformant. For that we have to do one understanding. I will show you one experiment where we will understand what is transformant, what is recombinant. Now here you do not know which are the selectable markers. I have to tell. Okay. So selectable markers can be your ampicillin, which is given here. Can you see? Ampicillin, tetracycline, kanamycin. All these are selectable markers which are present on the vectors. Okay, which are present on the vectors. So far good. So here if you see, students, don't you see selectable markers? Yes, you have ampicillin resistance and tetracycline. So PBr322 is an ideal vector because it has an ORI site. Okay, it has cloning site, it has selectable markers, right? All the things is connected, and it is also self-replicating. Now it becomes an ideal vector. Clear to all of you? Yes or no? Thumbs up in the chat if you understood. Yes. Students, with identifying I can also use the word elimination, right? Because they will help in elimination as well. So identifying and eliminating I will write, okay? So that it gives a better understanding of the concept. So it helps in identifying and eliminate. Identifying and eliminating transformant, non-transformant, recombinant, non-recombinant cells. Okay, and which are the selectable markers? You have ampicillin, tetracycline and kanamycin. Clear? Clear you. Very nice. Okay. Yes. Now we have to understand about the selectable markers, and there is one experiment. Okay, before that let's go for a 5 minutes break. Okay, because again my throat is the problem. So we will resume back. Okay, all of you come back. Don't go anywhere. Just stay in the live itself. Okay, I'll be back. Fine. Take a 2 minutes break. Just refresh and come back because we learned a lot of information, right? Fine. Yes, okay, I'm back. Are you all back? Okay, students, so far we have understood. Now I told you that vector should make sure that it does not have multiple recognition sites for one restriction enzyme, right? So here also same thing, again your cloning sites. So here you can see that it does not have multiple recognition site for one, for one restriction enzyme, right? Different ones are there, like 𝐂𝐴 you already studied. So here you have 𝐂𝐴. So you can understand that there is no multiple sites, okay? Multiple sites are not there, but one, at least a single site should be there, or it should not be more than one more site, okay? Too many recognition site for one restriction enzyme is not possible, okay? So vector should always make this. Now PBr322 is one of the, I'm writing it here. So your PBr322 is very important for you to remember, because your PBr322 is the first artificial, okay, it's the first artificial, okay, vector that was found. Okay, vector. Now this name does not come just like randomly, if someone did not make the name. Every letter has a meaning. Okay, every letter has a meaning. Students, share the tell your friends to come back. I think some went for break, means long break they'll take. Okay, so P stands for what, students? P stands for plasmid, obviously, right? Because plasmid. And B and R stands for the people who found it, that is your Bolivar and Rodriguez. Bolivar and Rodriguez are the people who found this plasmid, and 322 is the sequence with which, like, out of many plasmids that was found, the sequence number was 322. So this is your PBr322, and PBr322 is an ideal one because it has an ORI site, it has many cloning sites, it also has selectable markers. Now when we talk about selectable markers, okay, I think I have to inform. Okay, one second. Yes, done. Okay, so students, here see, when we talk about your PBr322, you have to understand what is the need for selectable markers. I already mentioned that it is helping for identifying and eliminating, identification as well as elimination of your non-transform and recombinant ones. Now let's take an experimental setup to understand this, which I have taught you last time also. We are going to take 3 cells, okay? This is the 3 cells, 1, 2 and 3 cells. These are the 3 cells that we have taken. Now, when I talk about the word recombinant, try to understand that when a plasmid or a vector with the desired gene has entered into the host, it is called as recombinant. What were we studying all this time? Recombinant DNA technology. That means a desired gene will be inside the host with the help of a vector. So when I am talking about the word recombinant, you are going to remember that, okay, vector is there with the desired gene. Now what is transformant? Let's say the vector has got transformed into the host cell without the desired gene, that will be called as your, that with or without the desired gene, that is called as transformed. That means vector has entered the host. Now host is no more normal, right? It has something has come additional. So same way here. So let's say first scenario, this is your genetic material and this is your vector. Okay, this is your vector without the desired gene. So here this is cell number A. You tell me what is this cell? This cell is a transformed, this cell is a transformed NR cell. That means non-recombinant cell. Why is it non-recombinant? Because it did not come with the desired gene. Correct? Am I right or wrong? Tell me if I am right or wrong. Okay, again. This is your plasmid, and in the plasmid there is the desired gene. Okay, desired gene is there. So can I call this B, okay, can I call this cell B as transformed, okay, or transformant and recombinant one. Okay, transformant and recombinant. Yes, ma'am, you can call it transformant and recombinant. Why? Because the desired gene has got into the host and also the vector is also inside the cell. Now, the third scenario, students, no transformation, no recombination, only the genetic material is there. So, this is going to be non-transformant. Non-transformant. Okay, this is cell number C. These are the three cells that we are going to see. Now, depending on the cells, you have to tell me. Let's take PBr322 we have taken. Now, selectable marker is amphicycline resistant and tetracycline resistant. That means what? They are resistant. Now, we have to come back. We'll come back to this after I explain this to all of you. Now, look here, students. When I'm talking about resistance, you should remember that. Let's say these are your, okay, this is a Petri plate. Okay, this is another Petri plate. This is going to have the growth of normal bacteria. Okay, these are your normal bacteria. Now, to the normal bacteria, okay, to this plate, let's say I am adding antibiotics. Okay, to this Petri plate, I am adding antibiotics. What is going to happen, students? If I added antibiotics to that plate, what is going to happen? Ma'am, definitely the bacteria is going to get killed. Bacteria killed. Why bacteria got killed? Because I added antibiotics. I added antibiotics. Now, same scenario we are going to repeat, okay? But I am going to grow. Okay, I am going to grow antibiotic resistant bacteria. Okay, I am going to grow antibiotic resistant bacteria. Now, what is going to happen? Same process I am going to continue. Okay, same process I will continue. Again, a Petri plate. This is your Petri plate. To this, again I am adding antibiotics. Okay, I am adding antibiotics. And what is shocking? I will see that nothing happened to the bacteria. All the bacteria are growing here. Why? Because they are antibiotic resistant. That means they can survive this situation. Right? They can survive because they are resistant to that antibiotic. Now, same thing is going to happen with your PBr322. We have amphicycline resistant and tetracycline resistant. So, if the vector has gotten and I have not done insertional inactivation. Now, ma'am, what is insertional inactivation? Let's say, students, insertional inactivation means, okay, insertional inactivation. From the term, what do you understand? I inserted something and something became inactive. Simple as that, right? I inserted some, knowledge into all of yours, then your doubts are invalid, right? It got inactivated. Same way, insertional inactivation means amphicycline resistant gene is there. With a restriction enzyme, I cut them and I put my desired gene here. Now, what will happen? Amphicycline resistant gene is going to become recombinant. That means I broke them. Okay, I broke them. Now, no more they are going to be resistant because I added a desired gene there. So, that is called as insertional inactivation. Because I inserted something, some portion of the selectable marker became inactive or that selectable marker became inactive. So, insertional inactivation in your textbook, you have two categories that you have to study. Insertional inactivation of your tetracycline or amphicycline, they can give you both, okay, amphicycline resistant gene. When insertional inactivation happened on tetracycline and amphicycline resistance in your PBr322 and your insertional inactivation with the help of beta galactosidase. Okay, galactosidase in PUC18, okay, PUC18. So, these are the two vectors that you have to study and you have to study insertional inactivation from it, okay. Now, if I write it that corner, you cannot see, no. 1 second, students. Are you all understanding? Sleepy? Okay. Are you understanding? Galactosidase in Paketi. Okay. Now, we have to learn this properly. Now, antibiotic resistance you understood, no? Now, let's say, students, this is a small portion. Okay, this is a small portion of your ampicillin resistant gene. Okay, ampicillin resistant gene. This is your ampicillin resistant gene on PBR322. I have taken it separately. Okay, I have taken it separately like this. Now, let's say I do insertional inactivation. I do what? Insertional inactivation. That means I insert something here. I insert my desired gene here. I insert my desired gene here. Now what is going to happen? This person who had, okay, this person who had, you know, activation, full active ampicillin resistance, I am you, grow me in ampicillin, nothing will happen to me, who was able to survive will no more be able to survive in ampicillin, right? Clear? So here, the moment I inserted the gene, here insertional inactivation happened. Insertional inactivation. Clear? Insertional inactivation happened. Now, let's go back here to PBR322 and let's understand, students, if I inserted something here, okay, near ampicillin resistance like this, okay, I inserted my gene here, ampicillin resistance will become inactive, correct? It will become inactive. Now, with that concept, if you understood insertional inactivation, let's go back to this experiment and try to understand. Now, imagine on the ampicillin resistant gene or on the, let's take tetracycline, okay, let's take tetracycline, insertional inactivation has happened, then I have to do a correction. I cannot draw the desired gene here, I have to draw it on this side, okay. So, everything that you draw is very important. If you are drawing this, you have to draw it on the correct side. Now, I have done insertional inactivation on tetracycline. Now, you tell me A cell, okay, let's take A cell, okay, this is your box, this is okay. Now, students, look here. First, I am going to write the cells, okay, like that. Okay, I am writing here ampicillin resistance, okay, and tetracycline resistance. Now, you are going to understand the actual use of your selectable marker. Okay, you are going to understand actual use of your selectable markers. If you understand this, it is going to be easy. So, let us say this is cell A, this is cell B, this is cell C. Okay, now you tell me, if I grow cell A, in ampicillin, A is going to grow or not in ampicillin? Just tell me yes or no. Okay, yes, I will put a tick if you tell me yes. Question is, if I grow it in ampicillin, will it grow or will it die? Okay, so is it a tick or is it a no? I have done insertional inactivation on tetracycline. Remember that insertional inactivation happened on tetracycline resistance gene, okay, on the selectable marker I have done. Let me see who is the smart one in the class who is going to answer fast. Who is going to answer fast? It's an easy question, nothing to worry, all you have to have is a little patience to answer. Very good, ma'am. It will definitely grow on ampicillin resistance because you did not do insertional inactivation there. Now tell me, will it grow on tetracycline? Will it grow on tetracycline? Answer quickly. It will grow in tetracycline or not? A, cell A. Okay, this is your cell A. A, B and C. Yes, it will grow. Very good. It will grow on tetracycline also. Now, what about cell B? It got transformed and here insertional inactivation happened. Will it grow on ampicillin? Will it grow on ampicillin? Quickly tell. Yes, very good. I don't know for which one you people are answering. Tell me, is it cell A or cell B? Cell A or cell B? Students, antibiotic resistance you understood, I hope. See, this is your antibiotic resistance. So that means ampicillin resistant and tetracycline resistant genes are there on it. That means they can survive. Okay, with this understanding, move forward. Okay, so biotechnology is all about understanding it properly, otherwise you will not. Okay, so yes ma'am, B cell is going to grow on ampicillin, but it is not going to grow on tetracycline. Why so? Because you did insertional inactivation. You did insertional inactivation and you made that gene recombinant. Now that is not going to act upon. Correct? Perfect? Is this clear? Those who did not understand, please raise your hands. Those who did not understand, raise your hands if this is not clear. Very good, Pooja. Very good. I can only see Pooja answering. Others? Okay, very good, Sneha. Hi, Jerusha. Yes, very good, Madhumita. Okay, Madhumati. So, understood this? That means here I damaged the selectable marker, and selectable markers are also for the purpose for identification and elimination. Okay. Now, what about cell C, students? Cell C. Cell C, will it grow in ampicillin resistant? Will it grow in tetracycline? Because it is non-transformant. Vector itself did not enter. Here what happened? Vector entered alone, forgot what is its job. It forgot the job and it just entered. Okay. Very late you became, Jerusha. Main topics all are done. Main topics, so many main topics are done. Yes. Clear all of you? So very simple, students. This is transformant and non-recombinant. That means only vector got in, no desired gene. So here there is no insertional inactivation. So it will grow both on ampicillin and tetracycline. Why? Because here we are talking about PBR322. PBR322 has two selectable marker. One is ampicillin resistance, like how I taught you antibiotic resistance. One is tetracycline resistant. That means they can grow in ampicillin and tetracycline medium. Then let's say B cell. B cell, what did I do? I did insertional inactivation on tetracycline. So it will grow in ampicillin. It will not grow in tetracycline. Okay. Non-transformant DNA you don't have to write because it will not survive here. It will not survive here also because no PBR322 only. Clear to all of you? This box is perfectly clear. Vandana, is it clear? So this is your how selectable markers are going to work. Now, because of selectable markers, I understood which is the cell that got recombinant and which are the cells which are just transformant. Transformant means where the vector has come in. Recombinant means where the vector has the desired gene also with it. Now, with this, I will be able to determine exactly which is the cell that got recombinant, which is the cell that got transformant. Okay? Perfectly okay to all of you? So this is the happiness of understanding concepts, okay? So understand the concepts and move forward. Otherwise, NEET will always be difficult. Yes, this is clear to my students, okay? This is also clear. Insertional inactivation also is clear. Now, yes, now we have to learn about one more insertional inactivation. This is clear, no? Transformant. Now we have beta galactosidase. That also we have to understand. Beta galactosidase. So students, your PUC18, okay? PUC18 plasmid is there. Okay, that PUC18, okay, first I will draw that. This is your PUC18 plasmid, okay, PUC18 plasmid. Now on the PUC18 plasmid, we have different regions, okay. Shall I just color it? Don't know how it will look. No need. So here two regions are there, students. One is going to be ampicillin resistant, okay, one selectable marker they have. One selectable marker they have, okay, and on the other side they have a set of cloning sites, okay. They have many cloning sites, many of them, okay, around 13 of them they have. Luckily, you don't have to remember the 13 of them, okay. 1 second, let me finish drawing. Yes. So look here, this is your PUC18. Okay, PUC18 we tell. Okay, that's nice to tell. No, it is found by Plasmid University. It's P for Plasmid, UC for University of California. Okay, what is it? PUC18. Okay, so as the name you know that it is P for Plasmid, UC for University of California, and number. Okay, sequence is there. Now here you have to remember insertional inactivation once again. Okay, now students, as I told you, this PUC18 has a lot of cloning sites. Okay, this is your PUC18. It has a lot of cloning sites, up to 13 sites. You do not have to remember the name. Now that when it has many cloning sites, we call it as multiple cloning site, MCS. So this portion which I have drawn in your orange color is an MCS. Why? Because many cloning site, be it ECO-R1, okay, be it HIND3. So let me just check, okay, all the sites, because in case I write it wrong, it will be, okay, SMA1. You don't have to remember this, I am just writing. Multiple cloning sites are there, so we call it MC, which is called as multiple cloning site. Now, most important thing is they have a lag Z. What do they have? A lag Z, which is your lag Z that you have studied in your molecular basis of inheritance. Now, lag Z in your lag coupron, which is the gene that, which is the enzyme that they form the enzyme beta galactosidase. What do they form? Beta galactosidase. Now, if I want to check, okay, if I want to check if lag Z or your lag Z is intact or recombinant, or if insertional inactivation has happened, I will use one technique. Okay, so I know that lag Z is going to form beta galactosidase. Okay, students, definitely they are going to have an ORI site. Okay, just because I didn't draw does not mean it does not have. It has an ORI site, that's why it's selected as a vector, right? Obviously ORI site is going to be there. Ampicillin resistance is there. Luckily you do not have to know much about it. Okay, this is going to be your ORI site. Okay, ORI site. Fine, ORI is there. One selectable marker is there, and many cloning sites are there. Okay, now this is your PUC18. Now in this, what you are supposed to remember? What you are supposed to remember, that if lag Z is intact, what is the scenario? What is going to happen? For that, I am going to take a substrate. Okay, I am going to take a substrate, and the substrate name is 𝐂𝐤. What is the substrate name? 𝐂𝐤. So students, all of you know that enzyme substrate complex, right? All of you know E plus S, right? Enzyme plus S, that is enzyme substrate, will form ES, which is an enzyme substrate complex that will ultimately give me a product. So here I have an enzyme, here I have given a substrate. What am I expecting? I am expecting a product, right? I am expecting a product. So if I am going to get a product, there is one way to do it. Now I am going to use a dye for this. Okay, I am going to use a chromogenic dye, which is going to tell me if lag Z is intact or lag Z got recombinant. Now what am I trying to say? That is, here I have done insertional inactivation of desired gene. Understood, all of you? That means here if I have put my genes, okay, here let's say if I put my desired genes here, what is going to happen? Lag Z is going to get inactivated and it is going to become recombinant, right? So to check that, I am going to use 𝐂𝐤. With the 𝐂𝐤, I am also going to use the dye. Is this clear, or is it too difficult to remember? Fine. Okay, fine to all of you know. Now, I am going to use a dye which is going to tell me if I got a product or not. Let's say if I get, okay, this is my Petri plate, okay, this is my Petri plate. On this Petri plate, let's say I got some white color colonies. I got, so instead of petri plate, I'll just draw, okay, normal plate like this. Okay, here, let's say I got some white colonies. Okay, and I got some blue colonies. Now you have to know what are these colors indicating. So the dye I used will give me, okay, I used a dye here. Dye is used. Now, the dye is going to give me a blue color if, okay, it is going to give me a blue color, blue colored colonies if my lag Z or lag Z is intact. Intact means what? Insertional inactivation did not happen. Insertional inactivation did not happen. And if I get white colonies, if I get white colonies, what does it indicate, students? That means my lag Z has got recombinant. Okay, lag Z has got recombinant. Now, what is this white or blue? Imagine this is your Puck 18, right? This one like this is blue color, okay? The other one is like this, white color. Many colonies of that will be forming, okay? Many colonies will come together. Yes. So, students, blue color means it is non-recombinant or lag Z is intact. White color means, that means your lag Z got recombinant. That means very simple that on the white colonies that you're seeing are the result of insertional inactivation. Insertional inactivation. Clear to all of you? Yes or no? Is this clear? How we got the white and the blue? Yes. I hope it was clear. If there is anyone who hasn't understood, please ask me the doubt and I'm going to tell it, okay? Don't hesitate. Don't be like, oh, you ma'am is not going to tell. Clear? Perfect. All of you understood now? Very nice. Okay, very nice. This was clear? So, this is the two insertional inactivation that you have studied. One is with your PBR322 where your tetracycline resistant gene will be insertional inactivation will happen on that selectable marker. Here on lag Z we will do an insertional inactivation. So, between the lag Z, if I have put a desired gene, I am going to get a white colored colony, and if I have not inserted, okay, that also I will just write it here. Okay, 1 second, students. And MCS is important for you to remember. It's a multiple cloning site. Okay, multiple cloning site. Okay, a lag Z is here and here, same thing. Okay, here lag Z is there, but between that I have done desired gene. Okay. Desired gene. Students, all these notes you will get it on Telegram, and if you want to join my Telegram, this is the Telegram. Okay, Biology by Gopika. Clear, ma'am? Clear, clear. Okay, so this is clear. Now, students, I'll teach you TI plasmid. Okay, TI plasmid I'll teach you. Then your plasmids will be almost done. Okay, TI plasmid. Now, TI plasmid, when I talk about it, is full form. What is the full form? Agrobacterium. It is from Agrobacterium tumefacium. Okay, Agrobacterium tumefacium. Okay. Now, Agrobacterium tumefacium, you have to remember that it enters into a dicot plant with the help of a wound. Now wound, how it happens? When you carve your names on the trees, when you like when you use a sharp object and remove the remove the first layer, okay, the bark you see, the black brown color, you remove that and you make an engraving or a mark on the tree. That is a wound on the tree, right? How you get a cut. Now you go fall somewhere, you get a cut, no, that is a cut, that's a wound. Same way on the trees also, if there is a wound present, students, let's say this is a wound present on the tree, like this wound won't be there, let's say that like this one wound is there, this is a wound. Through this wound, your Agrobacterium tumefacium will enter the plant. Okay, it will enter inside, and after it enters, what it is going to do? It is going to cause a crown gall disease. What disease? Crown gall disease. Basically, it is a swelling. Okay, it will be like a swelling. How does it look like? It looks like this. This you understood, no? Now, through the wound, it has entered the plant. It will cause a swelling like this. Okay, swelling like this. Completely infected. Don't get confused, okay, why ma'am is teaching this, okay. Oh, target done. Very nice, yeah, very nice. Today I didn't have to, okay, tell you people. Yes, it happens usually in a dicot plant, okay. It usually happens in a dicot plant. This is clear? Crown gall disease, this is a basic swelling. Now, why am I teaching about this? I went from, biotechnology, I went to disease, right? Okay, I went to disease. Why did I go to disease? Is because you will understand that there are some people who are really harmful for that. Okay, they are harmful for us. But if you remove that one screw that is loose, okay, one thing that is not okay about them, then they'll become good for us. Okay, like let's say someone doesn't like, they don't like us, they keep telling wrong things about us. Now if you make them keep quiet, then they are not a bad person. When they are silent, they are a good person. When they start talking only, they become a bad person. Same way here also, students, your TI plasmid, which is from your Agrobacterium tumefacium, is a wrong person, because through a wound it will enter into a plant, a dicot plant, and cause a crown gall disease which infects the entire plant. Now from that, okay, from that Agrobacterium tumefacium, I am going to remove one thing. Okay, I am going to remove the person who is the problematic one. That is, I am going to remove the tDNA which is actually causing the infection. tDNA which is actually causing the infection and affecting all the plants. So this is the tDNA which is present, which is causing the problem. Okay. What I will do? I will remove this tDNA who is the problematic one, and to that place I am going to add my desired gene. Problem sorted. A problematic fellow became a non-problematic fellow. How? Because I removed the tDNA and to that place I added my desired gene. I added my desired gene. Clear to all of you? Is this easy? Removed the tDNA and added my desired gene. Now when I added my desired gene, since it already has the ability to do cell division, and it is going to, students, by the way, I forgot to tell you that this crown gall disease is happening because of the continuous cell division. Okay, from this wound this got entered, no, after this will do continuous cell division. Remember, it will do continuous cell division, and it is going to, with the help your cytokinin and auxin. Okay, so these two people are going to be there. Yes, remove tDNA, add desired DNA. Okay, so it is going to do continuous cell division with the help of cytokinin and auxin. And what are they going to do? cytokinin and auxin. So we can use this technique, which is allowing us to make multiple copies. I want multiple copies. My vector should do the job of getting into the host and making, make sure that the host is competent. Vector should enter inside. So I made this the vector. What did I do? I removed the tDNA, who is problematic one, and there I added desired gene. Okay? Yes? Clear to all of you? Yes. Yes. No, Roshni, tumor means a mass of cell. Okay, a mass of cell can cause a lump. We tell, no, lump, a swelling. So, like that. Okay, two different terminologies. This so far good? Yes, clear, right? Okay, perfect. Okay, this is one TI plasmid where I remove the tDNA desired gene. Students, same thing we can do with one more problematic fellow which we studied recently, that is your retrovirus. Retrovirus have the technology, right, have the enzyme that can convert RNA, can go reverse of your central dogma, RNA to DNA. So what we will do, we will remove the problematic part, maybe we will remove the reverse transcriptase, or we will remove some problematic part, okay, because exactly it is not mentioned. So we will remove the problematic one and we will use this retrovirus as a plasmid, because they have good ability to multiply in the host cell. So we will also use retrovirus, okay, cancer causing cells also, to do this job of plasmid, like act like a plasmid, because they will work very good inside the host cell. Fine. Oh, is it? Results came now. All of you don't run away, wait for 2 minutes. Okay, don't run away like, oh my god, results came. What am I supposed to do? Done? Clear? Yes? Yes. Okay. Now, PCR is left, students. So there are different steps for your recombinant DNA technology, right? That is, your isolation is there, forming the fragments is there. Now you studied all of that, right? Fragment formation you studied. Now the host has to be competent. That means if I want to introduce this vector, right, my host have to be competent and it should be ready to accept this vector inside them. So for that I can do a heat shock, okay, I can do different techniques like your gene gun, okay, and also we can tell injections, okay, gene gun, your micro injections also we can do. That means where we directly insert the vector into the host. Clear to all of you? 35% scholarship. Wow, Saif, that's really good, yeah. Others, what about others? Oh my god, I'm excited to know about your this one. Okay, I'm excited to know about your results now. Tell me. Anyone else? Anyone else who got something? No? Yes, micro injection in animals, okay. Perfectly okay so far, no? Now, I'm thinking I'll teach you about the host and I think that's it, okay. Host I have to teach you. Yes. You can use that 35% to get into any offline center of Vedantu, okay. If you have an offline center near your home, definitely go there. That 35% will be reduced from your total fees and your parents are going to be super proud, right? They'll be like, okay, thank God my son has done this, okay. Students, so when I want to insert the vector into the host, I can do transformation, okay, transformation. That is, there can be pores, right? I can form the pores on the host and make sure that the vector enters inside, okay. For that, I can use divalent solutions, right? I will use divalent solution, which is your calcium chloride, right? Calcium chloride, CaCl2. Ca. Yes, we'll come to that. Okay, heat shock, micro injection, biolistics, and TI plasmid, retrovirus, will come into that. Okay, now is this clear to all of you? Now what is going to happen, students? Remember that your, we can add your host into this divalent solution, and the host will start forming, will have pores. Okay, that is because of your DNA being hydrophobic and hydrophilic tails, that changes is going to happen. Okay, that is, your DNA is hydrophilic, that loves water, and because of that we will be able to make these pores easily on the DNA. You don't have to know a lot of detail. Okay, pores on the DNA, and these pores, with this pores we can allow the vector to enter inside. Okay, this is one technique where we make pores, and through the pores the vector can enter inside. Okay, pores will be formed, and here your DNA is hydrophilic. Okay, hydrophilic. DNA is hydrophilic. In case somewhere question comes, just remember. Okay, and your bacterial cell will be hydrophobic. So like that, with that connection we will make the pores, and vector will enter inside. This is one method which is not stressed a lot. The other methods which are stressed are going to be your methods to make your host competent or allow the vector inside. First one is your heat shock. Heat shock. I basically taught the entire chapter, even though I told main concept. Okay, heat shock, micro injections, gene gun, okay, and your plasmids, that is actually disarmed pathogens. Okay, disarmed pathogens, that means they were pathogen but we disarmed it. Okay, that is, they are no more harmful to us. Disarmed pathogens, these are the 4 of it. Okay, Mangalore we don't have, dear. Organisms and population will be done by Baswa sir. Okay, students, now here what is heat shock? We keep them in extreme heat, then we keep it in ice. Again we keep it in extreme heat, we keep it in ice. So this is called as heat shock method. Okay, next is micro injection, where we inject it directly into the nucleus. Okay, directly we inject it into the host. Then gene gun, at a it's like how you have learned about your Thomson model, Rutherford model, where we the electric gun, like that. Same way tungsten will be bombarded. Because of the bombarding, it will directly enter into the host. Okay, and last one is disarmed pathogen, where you can use your TI plasmid as well as retrovirus, where we disarmed or removed the problematic one, and there we inserted a desired gene, and they will do the continuous process. Clear? Yes, I'll just write here disarmed TI plasmid. That's why I taught you that before. TI plasmid and your retrovirus. Okay, clear? This is your methods. Perfect. 42 degrees. Okay, someone asking? Yes. One line I can write. Okay, yes, gold and tungsten, both is there. Okay, so here heat shock, students, it will be kept at 42 degrees Celsius. Again it will be kept with the ice. Okay, this process is going to continue, and there is going to be a heat shock. Micro injection, obviously injection. Okay, it is going to be a vectorless method, where it is going to have injection directly to the nucleus. Okay, nucleus. Then you have gene gun, which is high velocity bombarding of tungsten and with your gold. Okay, high velocity, where you will use tungsten and gold. Okay, gold. And then disarmed, you already understood, no, pathogen. So I have just given one word, I have not gone into the depth. Okay, I have not gone into the depth. Is this clear? Now PCR, 3 things you have to remember. Day. Okay, in Tamil and all it is very common, no? Day. PCR is a method where we do amplification, okay. A small fragment of DNA we can make millions of copies out of it, right. Amplification we can do. So this technique has 3 words to remember, D-A-E, DAE, okay. Denaturing of the DNA, then you have annealing, and we have extension. This is your PCR, polymerase chain reaction, okay. No one else's result came, only few people's result came, I believe, right? Only few people's result came, or everyone's result came? Everyone's result would have come, no? I have to check, students. Once I finish the class, I will definitely check, because now I cannot check, no? So I will check after the class. After the class. Where did the results come, by the way? Tell me that. Okay, we are talking about MVSAT, okay? Those students who do not know, it was a scholarship exam conducted by Vedantu, and most of our students wrote it, okay? We are happy for that. Denaturing, annealing, yes, 50%. Oh, 50%, wow. Guys, if you are planning to join for coaching, this is the best time for you to join. Why? Because you can get that scholarship, okay, that scholarship in your offline centers. So definitely go and take the scholarship. Don't sit at home and be like, oh, no need, I don't want to go for offline centers. Go for offline centers now that you have it, okay. Now that you have the opportunity, please make it the thing to go. Okay. So students, this is also one line I will tell, okay, one line I will tell. What is it? Denaturing means I have to separate the 2 strands of DNA. So if you want a detailed video, you can please check Biotechnology part 2, lecture part 2, where I am teaching you PCR, okay. Basically, you have to separate the DNA strands. So we are going to do that. Separate the DNA, okay, fragments. That is called as denaturing. Okay, in your annealing situation, we are going to add the did I tell oligosaccharide by mistake? Okay, oligonucleotide, okay, oligonucleotide as a primer. So we get the free 3OH group, okay. And then we have extension where we will add, okay. And here students, here you have to remember one most important thing, that is because it is going to happen at different temperatures. Now since you are going to keep it in a PCR machine, you are not going to every 5 minutes open and see, oh, is the temperature correct, open and see. It's not cooking. You are not making a sambar, okay, at home, or a khichdi at home. It's a PCR machine, and it is going to be a continuous process where all the 3 processes is going to happen. But the catch here is how all the 3 processes is going to happen. All the 3 processes is going to happen because of the change in temperature. Let's say one is at 90 to 94, another one is at 72, and another one is at 65. So here what is happening, we only change the temperature. We do not play with the PCR machine, opening, closing, nothing like that is going to happen, okay. We change the temperature, and correctly denaturation, annealing and extension will happen. Now since it is going to happen at such a high temperature, we cannot use your normal DNA polymerase. So we are going to use Taq polymerase to make our new strands, right. Taq polymerase, which is extracted from Thermus aquatis, which is surviving, okay, which is surviving in hot spring, okay, in extreme conditions, okay. So Taq polymerase is something that you have to remember, and Thermus aquatis also you have to remember. Clear to all of you? Yes. So we will use Taq polymerase in the extension technique to continue the formation of the fragments, right. We have to add the new template, no. So oligonucleotide is going to act like a primer. So they are going to come, and then more extensions is going to happen. Clear? Yes, Thermus aquatis. So this is your PCR, okay. Again, I have not gone into complete depth of it, but remember that it is used for the process of amplification. It is one step of your recombinant DNA technology, okay. After we form fragments, we will get small pieces of DNA. That DNA has to be amplified, happens with 3-step DAE, day, okay, where you have denaturation, annealing and extension. Denaturation where we are separating the segments, annealing where we are adding the oligonucleotides, and then extension is where we are using Taq polymerase to form the new template, so we can make multiple copies out of it. Clear? Perfect. Now sometimes you will see the word forward primer and reverse primer, sorry, backward I am telling, forward primer, that comes here, students. Forward primer and reverse primer. So here one is going to go from this direction 5' to 3', and one is going to come from 3' to 5'. Okay, clear? So the word, if you see somewhere, don't faint and fall, oh my god, ma'am did not tell me. Okay, remember that there is forward primer and reverse primer. That is just for your annealing process, okay, because when we are adding the oligonucleotide who is going to act like the primer, it is important that we have a free OH group, okay. Yes. Yes, so polymerase, okay, oligonucleotide, forward reverse primer, yes, very good, okay, forward reverse primer. So forward reverse primer, remember, one is going forward, one is going reverse, okay, and they are going to give a free OH group so that the bonding can happen, which I taught you in molecular basis of inheritance. Fine, done, done. Any more doubts? If no doubts, that's it for today's class, okay, that's it for today's class. I think we have covered everything important. Hope we haven't missed anything. Okay, don't worry, even if we miss something, we will do revision again, but still I'm just telling, okay. So in case something is missed out, nothing is missed out as such. Obviously more in depth I have not done like last time how I did. Here I have given you an overview of the entire chapter and I have given you a revision of it, okay. All these notes you will get it on Vedantu NEET English Telegram, and as well as, yes, 30 cycles make 1 billion copies. The numbering, sometimes they will give you calculations on that, but very rarely I have seen, okay. Yes. So I hope this session was really helpful, students. It's important concepts of biotechnology principle and processes. I want all of you to share it with your friends and ask them to also watch the session. And I did not see comments on the last video. I don't know what happened. A lot of you told me, yes ma'am, we will comment, and all of you vanished. So I hope all of you will put your comments in this video as well. And all these diagrams are made for you. So I will be sending it to you on the Telegram channel. And any doubts, do let me know, okay. Yes, tell me the doubt, Vinayak, okay. No doubts, I hope everything is clear. Those who have doubts, definitely you can tell, okay. So EFB's definition is actually integration of, organisms. So where we are mixing, students, biotechnology, where we have technology as well as bio, right. So there your EFB is trying to tell that how we can use organisms, okay, we can use organisms to integrate something out of it according to our desirement, right. Like I am using a bacteria, I am putting the vector into it and I am getting my desired gene, okay. If you want the exact lines of it, let me open your NCERT. Yes, the integration of natural sciences and organisms, cells, parts, molecular analogues for our products and services. So it is basically a lot of complicated words together, right. So we cannot remember it, but do not worry, definition as such they will not ask. They will ask you EFB, they'll ask you the full form of European Federation, EFB full form, or among the words that they have given, just identify which is the word or which is the definition word matching to EFB. Clear? Vinayak, you do not have to by heart that. You are not writing your board exams, no? So it's fine. If you are writing your board exams also, very rarely they'll ask such complicated definitions, because the integration of organisms to get our, which has our molecular analogues, parts, to get our products and services is more complicated, right? It's more like a commerce definition. So I hope, students, this video was helpful, and now I want all of you to remember that on YouTube there is a link for those students who joined us newly. Let me just show that link to all of you, okay, because many students do not know about this. Yes, so students, this is the video you're watching, okay, this is the video you're watching. If you haven't like the video, and if you go to more, students, you can see here your batches which you want to take, you can definitely take, okay, whichever batches you want to take, you can definitely take. And here down the lane, if you go, you will be able to see pressure box, Okay, in this treasure box, all the video links are there. Okay, all the video links are there. When classes was taken, what and all types of classes was taken, all the YouTube video links are there. Most of the notes are updated, so here you can see all the notes are there. And here is the main part. You have PYQ. So here, look here students, see here you have PYQ. So biotechnology principles, if you go like this, slide all the way, you will be able to see NEET PYQ. So click on that and you will be starting the test. Okay, click on the test and see DNA fragments generated by restriction endonuclease in a chemical reaction can be separated. How can you separate DNA fragments that are generated by restriction endonuclease in a chemical reaction? I am going to answer electrophoresis. I am going to save and do next. Which of the following is not correctly matched for the organism and its cell wall degradation enzyme? See, we have bacteria. So when you talk about isolation of DNA, you know that they give different types, no, like bacteria, lysozyme, chitin, like that. So look here, plant cell cellulase, algae, methylase, fungi, chitinase. So which among this is wrong? I think I cannot see your chats, by the way. 1 second students, I'll check here. Tell me the answer for this, guys. Let me see how many of you know. It's a very simple question, nothing like complicated, but tell me. Yes, gel electrophoresis I have answered. This one. So like that, questions are there. Now if I read, see here they have given the colonies of recombinant bacteria appear white in contrast to the blue colonies of non-recombinant bacteria because of non-recombinant bacteria containing beta-galactosidase, insertional inactivation of alpha-galactosidase in non-recombinant bacteria, insertional inactivation of alpha-galactosidase in recombinant bacteria, inactivation of glycosid. So like this. So here you can see. Okay, now if I do end test. Okay, questions are easy. I want to attempt later. Yeah, I want to attempt later. Submit and leave. Now here they will show me my performance. See, okay, 1 I did incorrect, 3 I did correct. Okay, I think overall, yeah. So this is how my performance is. See how many attempts, percentage, everything will be here. Okay, attempts, percentage, everything will be there. Total and yours. Okay, no incorrect, all correct. Okay, 3 correct, 3 questions I answered, 3 are also correct, 0 incorrect. Okay, I was like, why did it go wrong? Yes, so this is the thing. So all of you can definitely try right now itself after the class, because I've taught you all the main concepts. I want all of you to solve this 10 questions or 20 questions, 27 questions are there. I want all of you to solve this and feel proud of yourself. Okay, feel proud of yourself and also make me also proud. Okay, yes. Okay, thank you so much. See you all in next class. I hope all of you enjoyed the session. See you all in next class. Tata, bye-bye.